today's topic is. So when I say denaturing gradient gels, the term itself tells you denaturation is there. So the factors that will denature the DNA, it could be some chemical or a temperature also can be a factor, right? So denaturing gradient gel could be with the help of chemical like urea, you can denature the DNA. And if it is temperature, then it is thermal denaturation. And either of these case, you can denature the DNA. Of course, these are the advancements of gel electrophoresis. So either a chemical gradient or temperature gradient is used to separate the samples. So separate the samples when, how, where. So when I'm doing electrophoresis, the DNA molecules which are present in a sample, the samples are loaded in the wells, and as they are negatively charged, they will migrate towards positive electrode. And separation mostly occurs due to size and sieving of the gel and the net charge that is going to help migration or mobility of the sample, right? So sieving effect is on account of the gel porosity, right? So separation occurs in electrophoresis. Now, when you are going to add additional parameters like denaturation conditions, maybe chemical or temperature, surely you are going to get something much more than what was possible to get as results by simple gel electrophoresis, right? So, for example, when you're talking about DGG, that is denaturing gel, gradient gel rather, I should say, DGGE, denaturing gradient gel electrophoresis, or temperature gradient gel electrophoresis. Both these methods are are you can say include the thing that DNA fragments, they need to be separated, and you have to compare them. So when you are going to denature, whether with chemical or with the thermal application, uh, the fragments of the same length but which differ in a sequence, single base sequence maybe, can be separated. And the separation is based on the altered, or you can say reduced, electrophoretic mobility of the partial melted double stranded DNA molecule. All right. So you have to develop a linear gradient of temperature and that of the mm, urea, for example, if you are going for DGG. So overall, both these techniques, they are going to make you analyze the sequences where they, if they are different, if they are varying, their electrophoretic mobilities under denaturing conditions will be even more intensely changed. Under electrophoretic conditions, the mobilities are differing on the molecular size, and that's how you separate the molecules. But further, if there are base changes or alterations in the sequence, they cannot be very well identified by simple electrophoresis. So additional dimension is added by adding denaturation conditions, so that the mobility is going to get changed if there is change in the sequence, if there is a mutation, if there is a variation. So we have to learn this technique in details today, right? So here I'm trying to share the slide. Fine. So when I say this, TGG and DGG, what are their applications? The very first, as we are talking about systematics of bacteriology and microbial diversity, the very first aim would be profiling the community complexity or identifying the unculturable bacteria to find out the population dynamics in a microbial community, right? Also, you can study differential gene expression in mixed populations. You can monitor the enrichment cultures. You can compare the DNA extraction methods. You can screen the clone libraries, right? There are many applications when it comes to TGG or DGG, that is temperature based or chemical based gradient that you are applying to gel electrophoresis. Before we go into the details about the technique, let's revise some basic some basic terminologies that talk about uh, uh, Tgge. Now here, as you can see in the on the screen, the base pairing, that is ATGC base pairing according to Watson-Crick model is shown. Thymine pairs with adenine with the help of two hydrogen bonds, and cytosine pairs with guanine with the help of three hydrogen bonds. Now, if I attempt to take the DNA in a sample, a Penrose tube as a sample, and expose it to increasing temperature, what would happen? These hydrogen bonds will start melting after a particular melting temperature, right? So as I have double stranded DNA, I have exposed it to increasing temperature. The increased temperature will start weakening the hydrogen bonds, and the hydrogen bonds will start breaking. As the hydrogen bonds break, the in the localized areas, in the specific regions, in the specific domains, the double stranded DNA is no longer double stranded, but it is unwoven, right? So as the temperature is increased, the content or the length of the DNA that is unwounded is going to increase. And how can I monitor it? I can very well monitor it with the help of a UV visible spectrophotometer. I can just monitor the absorbance at 260 to 80 nanometers, and I can get to know if the increase in the absorbance is there with increasing temperature. Then I get to know that DNA is undergoing unwinding. Right. So next is, I had mentioned in certain domains or at localized areas, the melting would start. These areas are called as melting domains. Generally, 50 to 300 nucleotide long regions these are, and all nucleotides in a given melting domain, they dissociate. And what is the way they will dissociate? All or no in this manner. That means it is going to be dependent on what is the sequence therein. So if it is GC-GC rich, then there are going to be 333 hydrogen bonds per pair, and it will be difficult to break as against that of the hydrogen bonds between 80 that we know. So depending on the sequence, the time required is going to be different, or the temperature required is going to be different. So when I say melting temperature, it is the temperature required to dissociate the double stranded DNA, but not fully. It is 50% of it in molten state, right? We know that. So Tm is the melting temperature, which is defined as the temperature at which 50% of the individual molecules are dissociated in the given melting domain, and remaining 50% as a double helical strand. Now, as we know, this TM is going to be dependent on individual DNA sequence and can be very much altered by a small change in the DNA sequence, including single nucleotide change, single base pair change also can give a variation in TM values. For example, replacement of an AT base pair that is having two hydrogen bonds by a GC base pair that is having three hydrogen bonds, the TM value is going to be changed. Not only that, the pair that is AT, if I take it other way, TA, again will show a variation in its melting temperature. So sensitivity with respect to temperature is really good, and so it can be used to see if there are any variations present in the DNA. But how? Let's go about it. So when I say TGG or DGG, the temperature gradient gel electrophoresis, and DGG is denaturing gradient gel electrophoresis, both these techniques, they are based on a similar principle. What is that? The electrophoretic mobility of the double stranded DNA fragments is going to get reduced by denaturation. But partial denaturation, I'm saying, it's not complete denaturation. If it is complete denatured, then both the strands will be separating out, and they will loop out in a random way, and their mobility is going to be totally different. So under electrophoretic conditions. So I repeat the principle: electrophoretic mobility of the double stranded DNA fragments is significantly reduced by their partial denaturation. Now these methods are really very sensitive. They are very comparatively easy, and but you need to optimize the process, you need to design the process. How, we will talk about it little later. But main advantage is high detection rate and specificity. It can be improved for heterozygote detection. It is very simple, and simple in terms of the steps, as well as you are not using something like radioactive material, so it is comparatively non-toxic method. Point mutations in genetic disorders can be very well diagnosed using this technique. However, there are certain limitations also, and there where comes the point, that is designing and optimizing the process. Because whenever you want to check the point mutation in a given single sequence, that sequence needs to be amplified. And to amplify an unknown sequence, there is a challenge that what primer is to be used. So design of a primer is very basic and very important challenge in this case. So difficult to design adequate PCR primer and set up the assay is one of the limitations. Now, when it comes to limitations about this method, uh, additionally, the problem is PCR fragment length can be amplified up to 500 nucleotides. Another thing is the problem with, if the sequences are GC rich, then there is going to be a difficulty in analyzing it with DGG. And of course, you need computer analysis systems for analysis of the PCR fragments. But once you choose the appropriate primers and the conditions for amplification and to run the temperature gradient, of course, temperature gradient is to be chosen and is going to be dependent on what is the length and complexity of that sequence. Right? It is not going to be a single value like that in PCR. Ki, denaturation temperature, renaturation temperature, extension temperature are fixed. It is not so. When you are going to run the denaturation electrophoresis, then the temperature gradient is to be selected specifically, and that needs to be optimized. It is to be designed and optimized. Right? But yes, in spite of all this, it is really a good technique for finding out the mutations and detection of known unknown mutations in the larger genes, and sensitivity also is really good. Now we go to the steps in this. The technique of temperature gradient gel electrophoresis was developed by Lerman and Anderson of Georgia. They had utilized beryllium oxide plate as a thermal diffuser. When you say temperature gradient, you can visualize the electrophoretic apparatus that you have used. So and it is a tank filled with buffer, and submarine is the gel. You load it, and that tank is fitted with the electrodes. That's it. But when you have to introduce thermal gradient, you have to have thermal diffuser, and that is a metal plate which is attached to it. We will see the design of the instrument also in the next slides. So when you are talking about the steps involved in, you have to prepare the gel, pour the gel, load the gel, which is very much like your process involved in agarogel electrophoresis or polyacrylamide gel electrophoresis, and then you have to adjust the voltage, and you have to leave the gel for running, right? Then you have to stain the gel and visualize. Staining methods are there, which are going to include like silver stain, or certain selective stains also are there other than silver. That we know, as we have learned agarogel electrophoresis to details. So the steps are very much parallel to what you do in agarogel electrophoresis. The only difference is that you have to uh, insert a metal diffuser, and which is going to generate the temperature gradient, right? So when it is about the schematic flow of the diagram of the process, diagrammatically we can show it as you have the cell, right, from which, or a sample from which you have extracted the DNA. You have the total DNA, you have to design the primers, then you have to run the PCR, you get the PCR products, and you have to go for gel electrophoresis. Right? The gel electrophoresis process, when done as a usual routine normal gel electrophoresis, this is what is the ladder, and these are the bands formed by the samples, specifically in the lane 1, lane 2, lane 3, as you see in this diagram. Right? But when you apply thermal denaturation component or the parameter in addition to gel electrophoresis, you can see the scope or the range of bands that are formed just by introducing the temperature gradient, right? Is it comparable to this? A wide variety of bands are seen, and that is going to reveal so many things about that DNA sample, right? It may be cultural diversity, it may be mutations in particular population, and so on and so forth. Further, you have to extract each band, purify it, and then you can go for checking its sequence and find out where it differs, right? So this is the schematic representation for temperature gradient gel electrophoresis. Now if I have to write the process, of course, as I said, I have to design the primers, then run the polymerase chain reaction, electrophoresis, but with linearly increasing gradient, temperature gradient. And if it is DGG, you have to go for concentration of denaturing agent like urea that you have to increase. That is to be little different. That in DGG you have to have a gel which is going to have changing or increasing concentration of denaturing agent like urea incorporated into the gel. That is the difference. So gel is to be specially designed when you are talking about DGG. But when it is TGG, that is thermal denaturation, you are talking about, that time the gel is very much similar to your simple method of casting the gel, right? So here, when you are going to run these gradient gels, nucleotide substitutions and other small changes in the DNA sequence, they can be very much visualized because the DNA that is going to have different mobilities will form bands at different positions. The positions may be varying little a bit, but variation is going to be there. So electrophoretic mobility of DNA fragments is going to differ, and it is going to differ according to whether the fragment is completely double helical or is having some molten domains like melting domains, or it is at what at how many locations it is molten. And of course, in this case, the highest melting temperature is of no use because it will separate both the strands. If it is very low, minimum, then and then only there is possibility to reveal the differences in the mobility of the molecules. So lowest Tm is of importance. But how I do and decide about all these parameters and to design and optimize the process. So melting map is a concept. Softwares are available for this. So these are the computer programs which can be used to design the primers for TGG and to design the gradient. And of course, one of the examples I have quoted over here, it is melt 87, but it is not the only one. You have melt 94 and a series of these are available, and you have to choose the computer program or the software that you can I mean, you have to exercise on it and then use it further. So experimentation is required to set up and design and optimize the process. All right. So these are the basic steps in the TGG or DGG experiment. So when I'm saying to summarize whatever I have said now till now, to say that whenever you are talking about conventional protein or nucleic acid electrophoresis, the molecules are going to get separated on the basis of their respective mobilities, which are governed by their size or charge, right? But when you are talking about TGG, a new parameter is added that is going to facilitate the separation to a better extent, and that is nothing but the melting behavior of the molecule. Melting behavior is governed by the sequence, right? Um, the primary sequence or secondary and tertiary structure of the molecule also is going to be important. Secondary tertiary structure, that is the folding. Primary is polynucleotide, then double stranded, whether it is folded or looped or circular like plasmid, all these things are going to be important. So temperature, salt concentration, pH, all these factors, they decide the secondary and tertiary structure of the molecule, we know. So that is how the buffer preparation is required, but it is very much parallel to your electrophoretic separation method. So during electrophoresis, sample are going to migrate along the temperature gradient. So as the temperature rises, the molecule starts to denature. So you are going to have the amplified fragments PCR amplified fragments. Maybe they are small stretches of about 500 base pair long, and they are double stranded molecules. At certain temperature, the DNA will start to melt, and you will get these structures which are going to be like, say, forks, right? As if it is replicating over there. The structure conformation is going to get altered, right? And as the conformation is changed, the mobility is going to get altered, because the randomness in the molecule increases, and it is going to alter its conformation. Altered conformation is going to alter its mobility under applied charge. So if it is molten, then it is going to be comparatively less organized, and its mobility is going to get reduced, right? Of course, the melting temperature is going to dependent on the base sequence. The DNA fragments of same size but having different sequences will be moving at different rates, because their melting temperature is going to vary even if there is a single base pair variation. So let's look into the further details. This is what is the concern I was talking about, that is how the double stranded DNA would be. And whenever there is a melting domain that opens up as melting temperature is achieved, right? So this is a schematic diagram that shows the electrophoretic mobility, and this is the direction of electrophoresis, and this is the direction for temperature gradient. You can see here it is said as cold and here it is warm. Of course, this is a relative term. You have to specify the specific temperature ranges, you have to adjust. And so when it is cold, the DNA is in its native conformation, double stranded strand. But as the melting temperature is attained, melting domains will start melting. And at the end, you will have single stranded DNA. But this is of no use, because single stranded DNA are going to be the slowest ones to move, because they are going to be very much random and will fold upon, and it will be very difficult to move through the pores in the gel. So this is how you can like visualize how temperature gradient is going to have an impact on DNA that is moving under electrophoresis. This is the schematic diagram that shows TGG instrument, very compact, portable, small instrument, very much like your gel electrophoresis instrument, only except that it has got a metal strip that is going to act as a thermal diffuser. So it is going to be electrically insulated metal plate, which is heated at one end and cooled at the other end. So thermostatic baths are used, and it's a simple horizontal gel electrophoresis instrument. Uh, and these DNA molecules can be separated using TGG according to size, shape, and thermal stability of their conformational transition. What is conformational transition? dsDNA starts melting as melting temperature of a particular domain is attained. That much part is only going to open up, right? So it can be further better visualized like this: complete denaturation, and this is here I'm showing partial denaturation. All right. So when I'm talking about these methods for denaturation of DNA, then where can we have the application of these methods, right? And how I'm going to see the results, that is another part, is it? So whenever I talk about these methods for mm, separation of DNA fragments using gradients, the important factors are the sequence of the DNA, the electrophoretic mobility of the DNA, the melting domains in the DNA, the melting state of the DNA fragments, right? So the electrophoretic mobility, when I say of the DNA fragment, is going to be different, and it will differ according to whether the fragment is completely double helical, or if one or more melting domains have dissociated, or if complete dissociation and you have two separated single stranded molecules are formed. So each of these states can be visualized. Of course, for that, you have two different types of TGG. One is parallel and another is perpendicular. We will come to that, but it is just to summarize whatever we have talked till now. we will summarize first and then we'll go to the next part of it. So, the electrophoretic mobility of double helical DNA fragment is not significantly altered by single nucleotide substitution, right? When we are doing simple electrophoresis, but when you are adding the thermal gradient, then the denaturation is going to affect the mobility. So, the PCR product contains, for example, a mixture of two DNA fragments that differ at a single position, right? And single position, matlab if you say a heterozygous point mutation you have, that means entire sequence is same, but only at one position, one base is changed. So heterozygous single nucleotide substitution you have, right? And it's a point mutation. So both the fragments will initially progress to the gel. Can you imagine? Uh, uh, they are running at the same speed because molecular size is same. Only there is variation in the one base in the sequence, right? So when the molecules reach that point where its temperature is temperature of the unit is equal to Tm of that particular domain. So at that time, that domain will open up. The molecule will be having loops in the structure. Won't be a stable double helical structure, but it will be having some some loops, fork structures in between, and because of this randomness, the molecule will start will get the resistance more, and its mobility will be reduced. So mobility is changing due to transition from a completely duplex or double helical conformation to partially denatured one. Right, it's clear. So dissociation of the first few melting domains generally will not show any dramatic result, right? Very long, very high reduction in the mobility cannot be attained because the fragment takes on a complex and branch conformation. Due to the strong sequence dependence of the melting temperature, the dissociation or branching, and that is going to lead to retardation or reduction of electrophoretic mobility is going to occur at different levels of the temperature gradient, and that is going to be associated with the what you say different factors. For example, it is what kind of base substitution is there, whether it is increasing the hydrogen bonds or it is reducing the hydrogen bonds. मतलब 80 से 80 को replace GC ने किया है क्या या GC को replace 80 ने किया है क्या. So TM value will be changing, right? So the temperature gradient is of course going to be dependent on the sequence mainly. When, for example, when you have two homoduplex molecules, that is they are exactly same, and there are two different heteroduplex molecules, and you attempt to what you say melt them and reassociate them, right? So dissociation temperature and reannealing of DNA fragments you are trying, and this can be done to find out what you can say the single point base substitutions. I'll try to share the slide. Right, just a second. Right, this is what I'm talking about. Right, so this is double stranded one. Now this is capital A indicates the homoduplex, and with wild type, and this small a indicates a homoduplex but with a mutant, right? Now, if I heat it at a particular temperature, say 90 degrees Celsius, all these four strands are going to get separated. And if I attempt to reanneal those, what will happen? What are the probabilities? A pairs with capital A, and the original homoduplex is formed. Possibility is that the small a, that is homoduplex with the mutant molecules, small a, it reanneals as the original one. But equal probability is that one strand of wild and one strand of mutant will anneal as you get these hybrid or heteroduplexes. Alright. Now the ratio of these molecules is 1:1. If you have one, this is one wild, one mutant. So this is original, and this is hetero. So this is homo, this is hetero. So 1:1 ratio you have. So when you are talking about denaturation after this, so after this, if you go for TGG, what will happen? So reannealing of these molecules are producing wild type, mutant, and a mixture of the two. Right now, if I go for TGE of these samples, what will happen? Don't you think that mobility of these four strands will be differing? Of course, it will be differing, but not here and here. It will be must be differing for these hybrid molecules, right? Because their melting temperature is going to be different, and you will be able to find out it, because the electrophoretic bands which are going to get formed after the process is done are going to be at different positions. It won't be as exactly same as it was earlier, right? So here, for example, I'm trying to get, yeah, this. So here I am having these original bands, but if they are varying at particular position, here are so many of them, and that is because so many forms are there. But if I'm talking about a case where there is a single variant, that is heteroduplex, so in place of this you can have another bond which is getting appearing somewhere, and that will tell you ki yes, it is because there is something different than the original wild and mutated strains, and where it is mutated, that can be further analyzed by sequencing, but it will form a separate band, right? And that band will be little behind, as it is undergone uh, uh, it has reduced mobility because its TM value was approached, and it has undergone melting. So this is how you are going to have applications in various fields, including disease diagnosis, to culture diversity, to strain, to find the total number of strains, to find out the uncultured bacteria and their sequences, so on and so forth. Now, as I have talked about, you can apply TGG in two different forms. One could be perpendicular and one could be parallel. So when we are saying perpendicular or parallel, it is with reference to the temperature gradient that is applied. So if I'm applying the temperature gradient perpendicular to the electrophoretic migration, I call it as perpendicular TGG. And exactly on similar lines, if I apply temperature gradient which is parallel to electrophoretic migration, I'll call it as parallel TGG. Now, when I'm saying parallel and perpendicular, why should I have these variations? This the electrophoretic gel that is formed and gel documentation picture I am seeing here. It is the parallel TGG, that is electrophoretic mobility and the temperature gradient are parallel to each other. This kind of pattern is going to get generated, which is very much like your electrophoretic pattern, and you will be able to find out what is the difference in the each sample, right, by comparing the position of the band, considering that all of them uh, are 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 going to be I mean, is one sample, you are going to get variation, that is, I'm sorry, here, as you have number of whales, you can load different samples, and one whale will represent one sample. So you can compare what is the variation as against that of the standard or the reference. So many whales you have, and so many samples you can load at a time when it is parallel TGG. But when you are going to go with perpendicular mode, it is single sample analysis. You have to remember these 2 points: single sample analysis, and how that single sample behaves with changing temperature. That profile of electrophoretic mobility will get generated, and you will get the patterns developed, right? This is to show you a pattern is getting developed. So some different patterns will develop, and that will tell you about the analysis of a single sample. So perpendicular mode or parallel mode, you are going to choose depending upon what is your requirement. For example, if I have to find out what is the variation in a single DNA fragment, like earlier case we talked about wild type and mutant, and just that is the only one which is I am going to compare with the wild type, right? Whether there is mutation or not. So that time I can see what kind of a temperature variation is sorry, temperature profile is going to affect the separation profile. So this is the separation profile. The same nucleic acid at different temperatures is going to give you different pattern in the electrophoretic gel. So it won't be like a very precise and uh, non-diffused perfect band as such that you see in common electrophoretic gel. It will give you a pattern, and that pattern is the result of effect of temperature on that and degree of denaturation. Right. So when you have to analyze for one sample, you can go with perpendicular. But when you want to compare a few samples with that of the standard, then you have to go with parallel TGG. Right. Now, when I'm saying applications of TGG, mutation analysis you can do, heteroduplex analysis you can do. Variations, when I say, then you can check for methylation